Hi, my name is Ali Gagnon, and here's just a general overview of the Calvin cycle before we jump into the step-by-step animations. So the Calvin cycle goes by several different names, including the light-independent reactions and the carbon reactions. And this process takes place in the stroma of the chloroplast, which is the space in the chloroplast that isn't thylakoids. So here's our thylakoids, and this is going to take place in this kind of blank place here called the stroma. And so the Calvin cycle is a cyclic process, and the end goal of the Calvin cycle is to produce G3P, a molecule that is used to make glucose, sucrose, or starch. In other words, when we talk about plants making their own foods and sugars, this is the process that does that. And so here's an overview of the entire Calvin cycle, which we will go through step by step. So there are three phases of the Calvin cycle. The first phase is called carboxylation, or carbon fixation, and Rubisco is the main enzyme catalyzing this phase. We start with three molecules of RUBP, which are each 5 carbon molecules. And remember, the Calvin cycle is a cycle, so these carbon molecules have been recycled from the previous round of the Calvin cycle. This phase also uses an input, in other words, it wasn't recycled, it had to come from somewhere else, of 3 molecules of carbon dioxide, which is taken in through the stomata of leaves. It also has an input of water, which is absorbed through the roots. As you can see in the diagram, here's what we started with. Over here are RUBP. We input carbon and water. And now we're going to let the Rubisco enzyme work its magic to rearrange the carbon enzyme, the carbon atoms. So that's Rubisco doing its thing. So notice that we had 18 carbon atoms to start with. And after Rubisco, our main enzyme catalyzing this process, rearranged the carbons, we still have 18 carbon atoms. They're just grouped differently now. So now we have 6 molecules of 3 carbon molecules, and each of them are called PGA. And that's all of the first phase. Let's move on. So for the second phase of the Calvin cycle, called the reduction phase, we're starting with those same 6 molecules of PGA from the previous phase. And now we're inputting 6 molecules of ATP and 6 molecules of NADPH. There we go. So 1 ATP and 1 NADPH molecule for each molecule of PGA. And remember, the ATP and NADPH molecules are inputs. As you can see here, they're not recycled. We have to take these from somewhere else, and in this case, the ATP and NADPH molecules come from the light reactions. So let's see what we do with those. So the 6 ATP donate 1 phosphorus each and are oxidized into 6 ADP. As you can see here, it's now ADP, not ATP, and they lost 1 P. And you can also see that here, the ATP changes to ADP and loses the phosphorus. The 6 NADPH each lose their hydrogen and are oxidized into 6 NAD+. As you can see here, it's now NAD+, and you can also see on the diagram the NADPH changed into NADP+. So ATP and NADPH were oxidized. The 6 PGA molecules are reduced. They're still each 3 carbon molecules, but they're now called G3P. And I've represented these redox reactions with color changes. You'll notice they look a little different than the previous slide. So like I said, the ADP and the NADPH will go to be recycled in the light reactions. They're kind of recharged into ATP and NADPH so that they can be used in the Calvin cycle again. And as we can see in the diagram, like I said, the ATP and NADPH were oxidized into ADP and NADP+. That's a lot of letters. And the 6 PGA molecules were reduced into 6 G3P molecules. So here's our PGA, and they were changed here. So let's see what happens to those. What do we do with them now? So what is going to happen to these molecules? Well, one of the G3P molecules will leave the Calvin cycle to form starches or sucrose. As you can see in the diagram, one of them is leaving the cycle to do that. The other 5 G3P molecules are going to continue on in the Calvin cycle to be recycled in the third phase. So that one leaves. Let's see what happens to the other 5. So here we are in the third and final phase of the Calvin cycle, called regeneration, and we're left with 5 3-carbon molecules, because remember, one of them left. And we need to regenerate, hence the name, we need to regenerate these molecules so that they can be recycled into the next round of the Calvin cycle. So essentially, we need to rearrange 5 3-carbon molecules into 3 5-carbon molecules, because the first phase of the Calvin cycle is going to start over with those 3 recycled 5-carbon molecules, if you can recall that picture from earlier in this video. And you can see this in the diagram above. We're going to be starting over, so we're regenerating. So how are we going to go about rearranging again? We have to input 3 ATP molecules for this process, which again, those ATP are obtained from the light reactions, so they're an input, they're not recycled. We have to get them from somewhere else. So the 3 ATP molecules were oxidized into 3 ADP molecules. As you can see, they lost their little p, and you can also see this in the diagram. It was input ATP, output ADP. And again, they will be recycled back into ATP via the light reactions. And the 5 3-carbon G3P molecules have been rearranged into 3 5-carbon molecules, now called RUBP, which should sound familiar because they're the same molecules that were in the first phase. And notice that there are still 15 carbon atoms like there were on the last slide. Now they've just been rearranged. And so that's the whole Calvin cycle. Now these 3 RUBP molecules will go on to be used in the first phase again and again and again, and each cycle will yield starch or sucrose for the plant. So that's all.